Hi everybody, and welcome to our module on hemoglobin. Let's start by talking about how oxygen is transported in our blood. There's a very tiny amount of oxygen that dissolves in our plasma, and the amount that's dissolved is determined by Henry's law, which you probably studied in a college chemistry class. Henry's law says that the partial pressure of oxygen times the solubility of oxygen in water will equal the amount that's dissolved in liquid like plasma. And this turns out to be a very tiny amount of the total oxygen in the blood. If we didn't have hemoglobin, we would have very little oxygen carrying capacity and we would never have evolved to become the complex organisms that humans are. Luckily, however, we evolved hemoglobin, which carries 98% of the oxygen in our plasma, and this allows us to deliver much more energy to cells of the body. Hemoglobin is made up of globin chains and heme. Globin chains are large globular proteins, and there are several different kinds that are used to make the molecules found in our red cells. There are alpha globin chains, beta chains, gamma chains, and delta chains. And all hemoglobin molecules are made up of 4 chains in 2 pairs. The other component of hemoglobin is heme, which is a molecule. It's not a peptide. This is its structure shown on the screen here. It's made up of nitrogens and carbons with an iron in the center. And this iron is what is going to bind oxygen. And this ring that surrounds the iron is called a porphyrin ring. There are 3 major types of hemoglobin found in the human body. Hemoglobin A is the adult hemoglobin. It's the most common form of hemoglobin found in adults. And it's made up of 2 alpha chains and 2 beta chains. Just like I told you before, all hemoglobin is 4 chains in 2 pairs. The second type is hemoglobin A2. This is also an adult type of hemoglobin, but it's less common than hemoglobin A. This makes up only about 2 to 3% of adult hemoglobin, and it's made up of 2 alpha and 2 delta chains. And then finally, hemoglobin F is the fetal type of hemoglobin. This is what is found in babies in the womb, and it's made up of 2 alphas and 2 gamma chains. Next, let's talk about oxygen hemoglobin dissociation curves, which are a classic topic in pulmonary physiology. Shown on this slide is an example of an oxygen hemoglobin dissociation curve. It plots the partial pressure of oxygen in the bloodstream along the x-axis, and along the y-axis is the percentage of hemoglobin binding sites for oxygen that are saturated with a molecule of oxygen. So for example, if every single oxygen binding site was filled with a molecule of oxygen, then we'd be up here at 100 on the y-axis, and if not a single binding site for oxygen were filled with an oxygen molecule, we'd be down here at 0. And remember, the partial pressure of oxygen is basically a measurement of the oxygen concentration in the bloodstream. So just like you would expect, as the oxygen concentration in the bloodstream goes up, more and more binding sites of hemoglobin become saturated. But here's the thing, it doesn't go up in a straight line like this. Instead, it forms this special curve known as an S-shaped curve. And for step 1 of your boards, you need to understand why this curve is S-shaped like this and not a straight line like this. So each molecule of hemoglobin has 4 heme moieties, and each heme moiety has an iron binding site. Therefore, each molecule of hemoglobin can bind 4 oxygens. And it turns out that the heme groups do not simultaneously bind oxygen. In other words, they don't bind 4 molecules of oxygen all at once. Instead, they bind one, and then they bind a second, and then a third, and then the fourth. They bind them one after another in series. And not only do they bind them in series, but when the first oxygen molecule binds to hemoglobin, this increases the affinity of hemoglobin to bind a second molecule. Then the second molecule increases the affinity for a third, and the third increases the affinity for a fourth. The result is that the affinity of hemoglobin to bind the final oxygen, the 4th oxygen, is 300 times greater than the affinity for the first. And this phenomenon is called positive cooperativity, and this is what makes the curve S-shaped and not a straight line like this. Hemoglobin is an example of an allosteric protein. The word allosteric means other site. So allosteric proteins are proteins where binding of a substrate at one site changes the likelihood of binding at other sites. Most allosteric proteins are multi-subunit proteins like hemoglobin, which is made up of 4 proteins. So hemoglobin is therefore an example of an allosteric structure, and the cooperativity we talked about in the last slide is an example of a positive allosteric effect. It's called positive because binding of one molecule makes another molecule more likely to bind. There are also negative allosteric effects where binding of one molecule makes binding of another less likely to occur. So now let's go back and take a closer look at what's happening in the body as it relates to the oxygen hemoglobin dissociation curve. In our lungs, the partial pressure of oxygen is about 100. And so if you look at this curve, in our lungs, you can see that the hemoglobin in red cells is going to become 100% saturated. So that's what happens in the lungs. All of the binding sites basically get occupied by molecules of oxygen. In the tissues of our body, the PO2 is much lower. It's about 40. If you take a sample of venous blood, which is blood that has just left the tissues, and measure the partial pressure of oxygen, 40 is a normal result. So what's happening according to this curve in the tissues is that oxygen is only going to be about 75% saturated or so. So what this means is that hemoglobin molecules move through our lungs and become fully saturated, And then when they get to the tissues, they unload oxygen down to about 75% saturated, and that difference in saturation represents the oxygen released to tissues for metabolism. Next, let's talk about the different formations that hemoglobin can assume. The 4 globin chains in every molecule of hemoglobin can become arranged in 2 different formations. One is called the taut form, the other is called the relaxed form. The taut form tends to release oxygen. This is the favored form of hemoglobin in tissues because it allows more release of oxygen for metabolism. The relaxed form tends to hold on to oxygen. This is the favored form in the lungs because it allows more binding of oxygen. So in our body, if you look at all the hemoglobin, you will find some in the taut form and some in the relaxed form. But the relative percentages of the taut and relaxed form can change by certain environmental conditions. And when this happens, it shifts the oxygen-hemoglobin dissociation curve. And this is a very important concept of pulmonary physiology. So the idea here is that the affinity of hemoglobin for oxygen can change. It's not fixed. Hemoglobin molecules are modified by the environment within the red cells. And when these molecules are modified, you can get either more or less of the taut or relaxed form. And when that happens, when you get more or less of the taut or relaxed form, that changes the dissociation curve. It can shift either to the right or the left depending on conditions. So if you look at the graph on the screen here, imagine that the blue is hemoglobin in its baseline state. If conditions are right, the curve can shift to the right, like this red one here, or to the left, like this green one here. And what's causing it to shift to the right or the left is that conditions within red cells are favoring either more of the taut form or more of the relaxed form. So let's start by talking about a rightward shift of the hemoglobin-oxygen dissociation curve. If the red here represented a normal curve, the blue dotted line here would represent what the curve can change to when there is a right shift of the oxygen-hemoglobin dissociation curve. So to understand the implications of a right shift, let's talk about hemoglobin under normal circumstances first. So under normal circumstances, the partial pressure of oxygen in the lungs is about 100, and this causes hemoglobin to be 100% saturated. When the molecules of hemoglobin get to the tissues, the PaO2 is much lower at 40, and this causes hemoglobin to fall to 75% saturated. So what our body is doing under normal circumstances is unloading hemoglobin from 100% saturated to 75% saturated. When the curve shifts to the right, we still have a PaO2 of 100 in the lungs. We are still fully saturating hemoglobin in the lungs. The partial pressure in the tissues is still 40, but because of the right shift, we have now decreased hemoglobin saturation down to 50. So the rightward shift allows hemoglobin to go from 100 to 50 instead of 100 to 75. And that represents a significant unloading of oxygen, and therefore the rightward shift really helps to deliver much more oxygen to tissues. And I've shown this down at the bottom here. Normally we go from 100 to 75%, a change of 25%. But if we right shift the curve, we could go from 100 to 50%, which is a delta of 50. That represents significant unloading of oxygen. And what's shown on this curve is the same thing I just showed you in that table. Don't get thrown by the curve. Basically what we've got here is in the lungs, we have a PaO2 of about 100. In the tissues, we have a PaO2 of about 40. As you can see, we're normally 100% saturated in the lungs no matter which curve we're operating on. Whether we are on the normal curve or the right shifted curve, we're still fully saturating hemoglobin in the lungs. However, which curve we're operating on makes a big difference when we get to the tissues. If we're on the normal red curve, we only get down to 75% saturated. That's a delta of 25. If we're operating on the blue curve, we get all the way down to 50, which is a delta of 50, and therefore we've unloaded much more oxygen. So you can just remember, right curve shifts lead to the release of O2 in the tissues. That's the idea behind this. Hemoglobin undergoes this kind of transformation in environmental circumstances where lots of O2 needs to be released to tissues. So what are causes of right shifts? Well, anything that is associated with rising metabolic activity where the tissues need more oxygen. So how can hemoglobin tell when tissues need more oxygen? Well, one way is if the CO2 level is going up. CO2 is produced by metabolism, so if there's lots of CO2 around, that will be sensed by hemoglobin, and it will change its form to have more of that taut form present, which tends to release O2, and that will cause the curve to shift to the right to look like the blue line here. Anytime carbon dioxide level goes up, the pH inside of red cells falls. I talk about this in the videos on respiratory acidosis. So therefore, low pH is a sign of high CO2, which is a sign of rising metabolic activity. And therefore, low pH is another thing that causes hemoglobin to shift to the right. And then finally, if the temperature goes up, that means there must be lots of metabolic activity around. And therefore, that will also cause a shift to the right of the hemoglobin oxygen dissociation curve. And then finally, let me mention a concept in pulmonary physiology known as the P50. The P50 is the point along the x-axis where hemoglobin is 50% saturated. Anytime the curve shifts to the right, that increases the P50. To understand this, let's look at the graph at the bottom right side of the screen. If we're operating on the red curve, the P50 occurs right here at a partial pressure of oxygen of about 25. If we shift to the blue dotted curve, the P50 occurs here, which is at a partial pressure of about 40, which is higher. So therefore, we can say that any rightward shift increases the P50, which is simply a number used to describe where hemoglobin is in terms of oxygen dissociation. And if you want a simple mnemonic to remember all these things with, just remember that the word right starts with R, which stands for release oxygen, and it ends with T, which stands for favors the taut form of hemoglobin. And if you understand rightward shifts of the hemoglobin dissociation curve, it's very easy to understand left shifts. They are the opposite. Shown on the slide is a normal curve represented by red, and the green dotted line is a leftward shift. This decreases the P50, and left shifts occur anytime there's lower metabolic activity. Remember, they're the opposite of right shift. So if the CO2 level falls, or the pH rises, or the temperature falls, all of these things will cause oxygen dissociation curves to shift to the left. And when the curve shifts to the left, it's less likely to release oxygen and more likely to hold on to oxygen. You can just remember this, that left curves latch on to